Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
To keep immune cells from reacting to inappropriate triggers or dragging out their response for too long, they're equipped with sundry molecular safeguards called immune checkpoints that nearby immune cells can flip on. As important as these checkpoints are, many tumors take advantage of them, throwing on these molecular brakes to dampen the anti-tumor immune response. A common form of immunotherapy called immune checkpoint blockade, or checkpoint inhibition, seeks to counter this kind of immunosuppression by physically blocking the immune checkpoint molecules so they can't relay inhibitory signals. But while this treatment is successful in some patients, it fails in many others, and scientists don't entirely know why, or more importantly, how to overcome the tumor's immunosuppression. The idea of tackling cancer with a brain parasite arose in the 1960s and 1970s when scientists observed that Toxoplasma infection boosted murine immune resistance to several infections and diseases, including cancers. In the decades since, evidence that Toxoplasma infections could aid in cancer treatment has been mounting. And a study published in November in the Journal for ImmunoTherapy of Cancer found that treating mice with a checkpoint inhibitor while injecting Toxoplasma directly into a tumor shrank both the injected tumor and tumors in other parts of the body better than when the mice were treated with checkpo. The findings are really intriguing, and potentially important down the road, because there's been a lot of interest in hype and commercialization of checkpoint blockade therapy, says David Bzik, an immunoparasitologist at Dartmouth's Geisel School of Medicine who has spent the past decade EXPL. He adds that these results may help make checkpoint inhibitor therapy more effective in patients who don't initially respond to it. But experts in the field, including the new study's authors, note that challenges remain, particularly with regards to actually implementing a live parasite-based therapy. I think you can use this system to establish principles that may lead to better therapies, says Christopher Hunter, an immunoparasitologist at the University of Pennsylvania who was not involved with the study. If you can understand what this parasite is doing that other immunotherapies aren't doing, then maybe you can leverage that knowledge to develop new anti-cancer therapies. A brain parasite to treat cancer. Toxoplasma gondii is a single-celled eukaryotic parasite that can only replicate inside host. It's often found in soil or water, shed from cat feces, or in meat from infected animals. Unlike some pathogens that only infect certain cell types within select organisms, Toxoplasma is a pretty ubiquitous parasite, says Pascale Guiton, a microbiologist who studies Toxoplasma at California State University, East Bay, and was not involved with the new study. It can infect virtually all warm-blooded animals, and can infect virtually all nucleated cells. After entering a host cell, the parasite forms a protective vacuole that houses the replicating parasites until they're ready to burst out of the host cell, killing it in the process, and infecting new cells to replicate in. Using a potentially dangerous parasite to fight cancer may seem like throwing a patient out of the frying pan and into the fire, but the idea of using pathogens to treat cancer dates back over a century. Around the turn of the 20th century, New York-based cancer surgeon William Coley made a surprising discovery while reading through old patient records. 7 years earlier, a terminal cancer patient had contracted a bacterial infection. By injecting patients with it, he could induce cancer-killing inflammation without risking infection with live pathogens. Over time, though, Coley's toxin fell out of favor with doctors as a cancer treatment. He adds that the advent of radiation as a cancer treatment quickly superseded Coley's toxin in popularity. But the general idea of boosting the immune system to fight cancer persisted. For instance, in 2016, Fearing's group demonstrated that nanoparticles packed with a killed plant pathogen called cowpea mosaic virus could suppress metastatic tumor growth in multiple mouse cancer models, including ovarian, colon, and breast cancer. And a therapy called T-VEC, based on an oncolytic virus injected into a tumor, is in use to treat advanced melanoma. One reason to look at Toxoplasma in addition to these other in situ vaccination contenders is the type of immune response it elicits. Pathogens come in different flavors, explains Hunter. We need different types of immunity to fight different types of infections. Unlike the type of immunity that's optimized for fighting bacteria or parasitic worms, the mechanisms the immune system uses to fight intracellular infections such as Toxoplasma are the same pathways you want to engage T. That's because the parasite needs its host to survive long enough for it to reach the stage in its life cycle where it's ready to be passed on. At the beginning of a Toxoplasma infection, the parasite exists in a quickly replicating form called a tachyzoite. After a few weeks, parasites that survive the initial burst of inflammation hide out in long-lived cell types like those in skeletal muscle and the brain. Occasionally a parasite-filled cyst will rupture, causing local inflammation, but otherwise, the parasite can survive in cyst form for the rest of the host's life. Then, if tissues containing the cysts are ingested by a new host, Toxoplasma can spread. If the host dies when the parasite is in the tachyzoite form, the parasite won't be transmitted, because this form dies in the digestive tract. It's only when the parasite is safely housed inside a cyst that Toxoplasma is stable enough to spread when its host is consumed. To ensure both its progeny and its host survive long enough, Toxoplasma actually reigns in its own replication during the early stages of infection by triggering a robust immune response, albeit not so robust that the tachyzoites are all destroyed. The parasites could grow normally in medium supplemented with uracil, but couldn't replicate in a mammalian host where the RNA base wasn't freely available to scavenge. In fact, just injecting T cells from infected, tumor-bearing mice were enough to significantly suppress tumor growth in other cancer-struck but uninfected mice. Other studies showed the parasite was also effective in mouse models of pancreatic cancer and melanoma. The researchers also found that when they gave the mice two tumors, treating the animals systemically with the checkpoint inhibitor but only injecting one of the tumors with the parasite, the other tumor still shrank. He notes that the unfortunate reality of cancer therapy is that many patients are not diagnosed until their cancer has already metastasized, making it nearly impossible to treat or remove all the tumors. But with this new observation that the dual therapy also attacks distal tumors in mice, it might be possible to target a primary tumor with a treatment that would also hit metastasized tumors. New understanding of metastasis could lead to better treatments. Despite some promising results, Fearing says he ended up shifting away from using Toxoplasma as an in situ cancer vaccine a few years ago because of a formidable technical hurdle. The parasites can be harvested out of the host cells and frozen for long-term storage, then thawed and revived in a flask of fresh cells, but it takes a few days for the parasite numbers to bounce back to a useful titer. We need something that we can take out of the freezer or the refrigerator or off the shelf and inject into the patient, he remembers them saying, not something that has to be recovered from live cell culture every few days. It's not impossible to make it work, he says, but it's also not practical from a clinical standpoint. Technical challenges aside, all of the researchers who spoke with the scientist were doubtful that a therapy involving injecting patients with a live parasite, especially one that is particularly dangerous in immunosuppressed patients, would ever take hold in the clinic, even with an attenuated versio. Instead, he says, the goal of their research is to figure out what exactly it is about Toxoplasma infection that was so useful at overcoming resistance to checkpoint inhibition and shrinking tumors. Rather than simply a pathogen, El-Shika sees Toxoplasma as a powerful tool to understand fundamental biological mechanisms. He wondered if Toxoplasma's ability to force target cells to change their normal biology could be harnessed and exploited in an innovative way. This is exactly what we have done in our study, he says. It's relatively easy to genetically manipulate compared to lots of other parasites. He says this makes it easy for immunologists and microbiologists to use the parasite as a tool to answer questions about the immune system, such as by knocking out certain genes to nail down which pathways are imp. For instance, he points to work his group has done showing how the cytokine interleukin 27, IL-27, inhibits the immune response during Toxoplasma infection, which has led to phase 1 clinical trial blocking IL-27 in advanced solid tumors. Additionally, understanding how the infection boosted the checkpoint blockade therapy is key from a cancer biology perspective, says Bzik. There's all these FDA-approved treatments using checkpoint blockade. For us to be able to explore these parasites for other ends like treating cancer, she says, we really need to study them and understand what's going on. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.